Hi everyone and welcome back to Show Academy. In this video, we're diving deep into Chapter 16, Chromosomes, Genes, and Protein Synthesis. This chapter is a high-frequency zone in Paper 2 MCQs. Questions from this chapter almost always show up, and if you understand the logic behind them, not just the definitions, you're guaranteed to score full marks. We'll break it down into three main topics. First is chromosomes, second is inheriting genes, and third is genes and protein synthesis. And we'll go step by step through every concept the examiner loves to test, with real examples, scenarios, and shortcuts for fast, confident answers. Let's begin with the first topic, the chromosome. The chromosome is defined as a thread-like structure of DNA, carrying genetic information in the form of genes. Know this definition well. Then, here are three important subtopics. First, chromosomes in haploid and diploid cells. What must be known here for the scope of Paper 2, it is a hotspot for MCQ questions, comes very often, and gives a lot of scenarios, all focused on the same facts. Number 1. Any body cell, even in the nose, eyes, or skin, contains the diploid number of chromosomes. So do zygote or embryo cells. All have the diploid number. Only gametes, like the sperm cell or ovum, contain a haploid nucleus, half the chromosome number. Number 2. A diploid human cell has 22 pairs of chromosomes and 2 sex chromosomes, or 1 pair of SE chromosomes. Second, mitosis. Number 1. Mitosis is nuclear division that produces 2 genetically identical cells, important for growth and repair. Number 2. You must know what happens to the mass of DNA in a nucleus during mitosis. DNA mass first doubles before mitosis, then halves during mitosis, to ensure each daughter cell receives a full set. Number 3. Mitosis maintains chromosome number. Number 4. Functions specific to mitosis include growth, tissue repair, replacement, and asexual reproduction, not gamete production. Number 5. The mitotic index of a tissue is the percentage of cells in the tissue that are dividing by mitosis. The tissue with the highest mitotic index is growing the fastest. Third, meiosis. You must know the following facts. Number 1. Meiosis produces 4 genetically different daughter cells. Number 2. Meiosis halves the chromosome number. It produces gametes. Number 3. Meiosis allows new combinations of chromosomes, promoting genetic variation. Number 4. Features like genetic variation, gamete production, and reduction division are associated with meiosis. Now let's move on to the second topic, inheriting genes. There are several subtopics here, and we'll go through them one by one, focusing on what usually comes in Paper 2 MCQs. First, genes and alleles. A gene is a length of DNA that codes for a protein. Alleles are different versions of the same gene. A mutation is a change in a gene that may lead to a different protein. Second, genotype and phenotype. The phenotype is the observable features of an organism, things like eye color, blood group, or hair type. The genotype is the actual genetic makeup, the alleles that are present in the DNA. Subtopic 3. Dominant and recessive alleles. A dominant allele is always expressed in the phenotype, even if there's only one copy. A recessive allele is only expressed if both alleles are recessive, meaning the individual is homozygous recessive. In exams, the focus isn't on definitions, but on how these alleles behave in genetic crosses. Most questions fall into one of 4 scenarios. Scenario 1. The offspring shows a phenotype that neither parent shows. This means the trait is recessive, and both parents must be heterozygous carriers. They carry the recessive allele but don't show it themselves. This is a classic example of recessive inheritance. The trait skips a generation and reappears in the child. For example, if a child has a recessive condition like albinism or cystic fibrosis, but both parents appear normal, that means both parents must be carriers. Their genotype is heterozygous. See this question. They ask about individuals who are heterozygous for albinism. To produce a child with albinism, both parents, for example individuals 5 and 6, must be heterozygous. And of course, individual 7, who shows the condition while both parents don't, confirms this scenario. Scenario 2. The offspring shows the same phenotype as only one of the parents. This suggests the trait is dominant, because just one copy of the dominant allele was enough for it to appear. The child likely inherited the dominant allele from the affected parent, while the other parent is homozygous recessive. This pattern appears clearly in this question. You see the phenotype polydactyly appear in the child, even though only one parent shows it. That's strong evidence that the trait is dominant. Scenario 3. Two individuals with the same phenotype produce 100% offspring of the same phenotype. In this case, the trait is either homozygous, either dominant or recessive. Both are still possible. To confirm recessiveness, you need other crosses to show variations. For example, in this question, if the examiner give you information that 2 orange-eyed flies always produce orange-eyed offspring, so you can extract a fact from here. Both parent are homozygous, but either orange eye is dominant or recessive allele. It will give the same result, 100% offspring, the same as parents. But when I tell you that 2 red-eyed flies can produce red or orange offspring, this tells you that orange is recessive and red is dominant. So if I only see offspring with the same phenotype, I cannot confirm recessiveness unless I'm also told how the trait behaves in other crosses. Scenario 4. You get a 1-to-1 phenotypic ratio in the offspring. This is a strong clue that one parent is heterozygous and the other is homozygous recessive. Half the offspring get 1 dominant allele and show the trait, and the other half get 2 recessive alleles and don't show it. This kind of cross is called a test cross. It's used to find out whether the dominant-looking parent is heterozygous or not. Look at this question, for example. The phenotypes among offspring are close to 50% dominant and 50% recessive. That only makes sense if one parent is heterozygous and the other is homozygous recessive. So the correct answer here is option C. If you can recognize which scenario you're looking at, you'll know how to answer the question. That's what the examiner is testing, your understanding of inheritance, not just definitions. Subtopic 4, Codominance. Codominance means both alleles are equally expressed in the phenotype. There is no intermediate phenotype. Both traits appear side by side, not blended. Key examples you must know. Red and white flower alleles give pink flowers. Roan cattle or horses show both red and white hairs together. A B blood group shows codominance between A and B alleles. If the question says no intermediate phenotype, like this question about blood grouping that also means codominance, don't hesitate. That phrase is just another way of saying both alleles are fully visible. That's what you need to know for codominance. Subtopic 5. Alleles in gametes and fertilization. Genetic information is passed from one generation to the next through gametes, sperm and egg cells. Each gamete carries only one of each kind of chromosome, which means just one allele per gene. That's why you must be able to work out the possible gametes produced by each parent, and what happens when those gametes join during fertilization. For example, if the genotype of one parent is IAIA, all the gametes from that parent will carry IA. If the other parent has genotype IAIO, then that parent can produce 2 types of gametes, one with IA and one with IO. During fertilization, any of these gametes can combine. So in this case, the offspring can have either IAIA or IAIO, both showing blood group A. This type of question is very common. They give you the genotypes of both parents and ask what genotypes or blood groups the offspring could have. You won't get it right unless you can correctly list the gametes and then match them through fertilization. So the key skill here is knowing what gametes each parent can produce and what combinations those gametes can make in the offspring. Now let's go through the different genetic crosses and their probabilities. This is a very frequent question in MCQs, and mastering it gives you a big advantage. First, if homozygous dominant crossed with homozygous dominant, all offspring will be homozygous dominant. That means 100% dominant phenotype. Second, homozygous recessive crossed with homozygous recessive. All offspring will be homozygous recessive. so 100% recessive phenotype. Third, homozygous dominant crossed with homozygous recessive. All offspring will be heterozygous. That's 100% dominant phenotype. Fourth, heterozygous crossed with homozygous dominant. Offspring will be 50% homozygous dominant and 50% heterozygous. So again, 100% dominant phenotype. Fifth, heterozygous crossed with homozygous recessive. Offspring, 50% heterozygous, 50% homozygous recessive. That's 50% dominant and 50% recessive phenotype. Sixth, heterozygous crossed with heterozygous, AA times AA. Offspring, 25% homozygous dominant, 50% heterozygous, and 25% homozygous recessive. So the phenotype ratio is 75% dominant, 25% recessive. Now let's look at codominants. Roan cattle or horses, roan crossed with roan. Offspring, 25% red, 50% roan, and 25% white. This is classic codominance. Both alleles are fully expressed. Finally, a common multiple allele example. Blood group cross, AB times AO. Offspring genotypes can be AA, AB, AO, or BO. That gives 50% blood group A, 25% AB, and 25% B. These cross patterns appear again and again in Paper 2 MCQs. Without memorizing, know the genotypes, write down the possible gametes, combine the gametes, and count outcomes with confidence. Then the sex linkage. Sex linkage refers to genes carried on the sex chromosomes, usually the X chromosome. A sex-linked condition is one where the gene is located on the X chromosome. The most common examples are red-green color blindness and hemophilia. Now let's go through the key inheritance patterns. If a carrier female marries an unaffected male, there's a 25% chance of affected sons and 25% chance of carrier daughters. If a carrier female marries an affected male, there's a 25% chance of affected sons and 25% chance of affected daughters. If an unaffected female marries an affected male, all sons will be healthy and all daughters will be carriers. If an affected female marries an unaffected male, all sons will be affected and all daughters will be carriers. And finally, if both parents are affected, all children, boys and girls, will be affected. These are the classic patterns tested in MCQs. Focus on who gives which X chromosome, and remember, X-linked recessive only needs one faulty X to show in males, so they're more vulnerable to X-linked diseases. Topic 3. Genes and protein synthesis. To understand how proteins are made, you need to know 4 key structures involved in the process. Number 1, DNA, which stays in the nucleus. Number 2, messenger RNA, which moves out to the cytoplasm. Number 3, ribosomes, where proteins are built. And number 4, amino acids, which float freely in the cytoplasm. Now let's go through the correct order of protein synthesis. First, the gene coding for the protein is copied inside the nucleus. This copy is called messenger RNA. Then, the messenger RNA leaves the nucleus and enters the cytoplasm. Next, the mRNA moves through a ribosome. As it passes through, amino acids are joined together in the correct order to form a protein. So, the genetic code in DNA, made up of the bases A, T, C, and G, determines the sequence of amino acids in the final protein. The role of mRNA is to carry this genetic message out of the nucleus and into the cytoplasm, where the protein is built. And finally, for protein synthesis to happen, you need both mRNA and amino acids in the cytoplasm. This is a key topic and may appear in MCQs or short-structured questions, so be confident with the sequence and roles of each part. And that wraps up everything you need to master from Chapter 16. Remember, don't just memorize, understand the patterns, work out probabilities, and apply the logic we covered. For more chapters, classified MCQs, and smart revision strategies, make sure to check out the full playlist and test yourself using our digital quiz linked in the comments. Stay sharp and keep scoring. Good luck.